Hello everyone, welcome to our November Hive-side Chat. I'd like to introduce our guest speaker, Liz Walsh. Liz has been beekeeping for around 13 years. Her first contact with beekeeping was as a high school student when she worked for a beekeeping supply company in Wisconsin, US. And she has been a beekeeping addict ever since, which I'm not surprised because bees are pretty awesome, right? Liz got her PhD doing honeybee queen research in Dr. Juliana Hangel's lab at Texas A&M University, and she's now the new postdoctoral fellow in Dr. Steve Purnell's lab at the Beaverlodge Research Farm. So I'd like to thank you, Liz, for your time today and for sharing your research results with us. Thank you.
Like the Varroa mite, which is pictured here in the top right-hand corner. So let's talk about Varroa mites for a second. So Varroa mites are the single most significant driver of honeybee mortality in North America. And part of that's because, as you can see on this picture, they predate on both immature honeybees, but also mature honeybees. They vector several viruses while feeding on their hosts, and they will ultimately kill a honeybee colony within about 2 years if they're left to go unchecked. And so as beekeepers, we treat Varroa infestations with extreme prejudice, right? And so one of the things that my dissertation research focused on was instead of agrochemical pesticides like the ones pictured here in the top left-hand corner, when we think of agro-applied pesticides, we commonly think of row crop applied pesticides that foragers will bring back to the colony. But in a 2010 survey of North American apiaries, what we found is that agriculturally applied pesticides like that applied to crops really only account for, in this case, 4 of the top 9 pesticides found in beeswax. The other 5 pesticides that were found in very high numbers and very high frequency are miticides that we as beekeepers introduce to our colonies in order to keep Varroa mites in check. And as you can see here, they're found at shockingly high frequency in these wax samples. This study was done by Mullen et al., and they found out of 251 colonies, fluvalinate and coumaphos, and let's just talk about fluvalinate and coumaphos for a sec. So the first 2 legal to use miticides were found in about 98% of all colonies that they sampled, which is shocking. Now, just a reminder about honeybee biology. Honeybees are eusocial. So what this means is that they exhibit cooperative brood care, overlapping generations, and a reproductive division of labor. And I'm sorry if I'm giving you whiplash going to bee biology, but keep the other stuff in mind for a second, and I will swing it back around so it all ties together, I promise. So the key hallmark of eusociality that matters to my research was the reproductive division of labor. Now, the female caste system in honeybees is reinforced in a variety of ways. It's reinforced through behavioral controls like worker-to-worker policing, where bees that are inside the colony, commonly nurses, will actually consume the eggs of any other workers that have attempted to lay eggs. we've also got pheromonal controls that reinforce this Caste system. And the pheromonal controls can be broken into two different components. Primer effects of pheromones. So up here in this top picture, you have the very, very well-developed ovaries of a queen. And on the right, you have worker ovaries. Here in the top right, you have the ovaries of a worker in a queen right colony. But in the bottom, you have the ovaries of a worker in a queenless colony. And you can see that there's a difference. This is because the pheromones that queens secrete act as this primer control, if you will, where ovaries in workers are suppressed through a very complicated hormonal cascades mostly. You also have releaser effects of pheromones, and an example of that is shown in the picture on the bottom. So here we have a queen in the middle, and her pheromones have caused a behavioral response, which is the releaser effect. And so these nurse-aged bees, are forming a retinue around the queen. And so they're caring for her. And that's one example of a releaser effect. Now, as beekeepers, we say all the time that a good queen is important. And the reason we say that is because queen quality is known to mediate colony physiology. This is just a scientific way of saying that if you have a good queen, you're likely to have a strong, good colony. But if you have a weak queen, you're a lot less likely to have a strong colony, right? So pheromones, queen pheromones at least, have been used as a proxy to indicate queen condition. So this is because pheromones in this particular instance are honest cues of queen health to workers who are the receivers of these pheromones. As such, queens attract their retinue with these pheromones, and queens that are healthy, that have a robust, attractive pheromonal mixture will attract large retinues, like in this top picture. Whereas queens that are less desirable, less healthy, will attract not as numerous retinues, like in the bottom picture. We know that the main queen pheromone, which is called QMP, or queen mandibular pheromone, is composed of fatty acid chains in addition to a few other compounds. And we're not going to get into the chemistry tonight. I just want you to know that we know what makes up these pheromones, and we can identify it and we can quantify it. Now, thank you very much for listening to the background. This is where it starts tying together. So the driving question of my dissertation was, how does pesticide exposure during development affect honeybee queen health in general? And tonight I'm going to talk to you about what ended up being two different chapters of my dissertation. The first of which was centered on queen pheromones and egg-laying rates, and the second of which was centered on sperm viability. And I hope you covered any children's eyes, because that might be the sexiest picture you've seen all day. But also queen mating frequency, so how many drones the queens mated with. So let's dive into the queen pheromonal stuff and egg-laying rates first. So the specific driving question of this work was, does pesticide exposure during queen development affect queen pheromones, specifically QMP? And if so, can workers notice? We suspected that the queen pheromones would be changed by the queen rearing environment, and that the workers would differentially attend queens that had been reared in different pesticide contaminated environments because of their different pheromone composition. So to do this, I got to learn a new skill. I had never grafted queens before I began my dissertation. And here's a picture of me grafting. I just used the JZ-BZ cups that I coated in wax. And this beeswax was certified pesticide free for the control group of queens, or it contained field relevant amounts of pesticides that were found in the Mullen et al. Study, which I neglected to mention earlier. The Mullen et al. Findings regarding pesticides have since been replicated. They've been found again over and over. And this has happened in a study by Kirsten Trainer, who was a bee field day visitor, and also by Nancy Osteguy. So in the decades since those findings were replicated. So once the queens that had been reared in these groups were mature and mated, I randomly introduced them to queenless observation colonies, such as the one shown here on the left. And then I point sampled by counting the workers in the queen's retinue for 5 consecutive minutes, many, many times. And so just to give you an idea, remember we talked about the queen retinue before. So I would go through and I would be like, okay, there's 1, there's 2, there's 3, there's 4, etc. For hours and hours. And I had a mix of queens that had both very robust retinues and weaker retinues. While I was taking this retinue data, I also counted the number of eggs each queen laid per minute for 5 consecutive minutes. So I point sampled that as well. And here are the first results. So I found that, so I'm not sure how familiar you are with statistics. So let's walk through this graph for a second. On the x-axis here, we have the treatment group. So the control queens that were reared in pesticide-free beeswax, and then the 3 experimental queen groups. And so fluvalinate and coumaphos are 2 miticides that were used historically. Amitraz we still use today. And chlorothalonil and chlorpyrifos were 2 pesticides that were found in conjunction, but those are agriculturally applied. On the y-axis, we have the number of bees in each queen's retinue per minute. And so the letters on top of the bars denote a significant difference in the retinue size. And so we see that control queens had significantly larger retinues than queens reared in any of the pesticide groups. More fascinating to me as a beekeeper, and also more alarming, was the egg laying rate data. So again, on the x-axis, we have the queen treatment group type. And this time on the y-axis, we have the average number of eggs laid per 5-minute interval. And here we see that control queens and queens reared in the agropesticides both laid significantly more eggs in a 5-minute interval than queens reared in either of miticide-laden groups. And honestly, almost twice as many eggs if you compare the control queen egg laying rate to the queens reared in amitraz, their egg laying rates. Okay, so the next step, because remember, I was interested in finding out if the retinue sizes were different, but also if the retinue sizes were different, if this was because of the queen pheromones. So then I got to figure out how to dissect queen mandibular glands, which are where QMP is generated. in a queen's body. So here, this is a picture of a queen head pinned to a dissection plate. And this sort of cafe au lait blob here is a mandibular gland. Each queen has 2. To give you a different perspective, here is that same mandibular gland with my finger for some sort of a size context. So they're quite small. So once I had the retinue data, I decapitated the queens, dissected their mandibular glands, and made sort of a mandibular gland extract. Right around the same time I was doing this, I collected emerging workers and allowed them to get to 5 days post-emergence. And here's a picture of me with the incubator bees. After the bees were about 5 days old, they were given 2 slides. One slide would have the pheromone extract of the control queens, and one slide would have the pheromonal extract of queens reared in one of the experimental groups. And here are those results. So again, along the y-axis, we have treatment groups or queen types, but this time we have them paired, so the control and a pesticide group. On the y-axis, we have the number of bees that were attracted to each slide per 5-minute period. And in this case, the asterisks indicate statistical significance. Here we see that the control queen extract attracted many, many more bees than the fluvalinate and coumaphos queen extract. And to a lesser extent, this is also true for the control queens versus the amatraz queens. This was not true for the control queens and the agrochemicals, and the queens reared in agrochemicals. All right, so after that had been done, I took the other mandibular gland because, again, each queen has 2 mandibular glands. So I did the choice test with 1 mandibular gland per queen, and I did GCMS analysis with the other. And GCMS is just gas chromatography and mass spectrometry, and it's just a way to analyze the compounds in a given substance. It's just a machine, which is pictured here, part of which is pictured here. This is a chromatograph. I'm not expecting you to read this. I just want to give you an example of what that output looks like. So here, each spike corresponds to a compound that is present in high amounts. And by comparing these spikes and the ion patterns to a library of compounds, you can identify what these are. And so in this case, they're all known queen pheromone components, which is really, I don't know, that's still exciting to me. So when I did some statistical magic, a principal component analysis, I was able to separate out which compounds in the queen mandibular gland pheromone were driving changes between the treatment groups. So here we have a very different graph, but what I want to show you is just that although there are no letters or asterisks, the sort of pheromonal signature of the control queen mandibular glands, shown in green, was statistically different from the pheromonal signature of the fluvalinate and coumaphos queens. But this was not the case for the queens in the agrochemical group or the amitraz group. And you can see this because the diamonds here overlap, and the control diamond and the fluvalinate and cumophos diamond do not overlap. So what does all of this mean? Well, we can see that the mandibular gland contents of queens were affected by pesticides in the rearing environment, and that these differences were so dramatic that workers could tell the difference. Some really fascinating work done by Princeton et al. In 2019 showed that queens exhibit pheromone redundancy. So queens have several different pheromones and several different glands that produce pheromones. And so I'm not sure if you noticed, but the differences in the retinue size were dramatic. The differences in the choice test were a little bit less dramatic depending on which comparison you were looking at. The differences in the actual pheromonal signatures, specifically of QMP, were different, but not as dramatic as the other two experiments. This is probably because the queens are exhibiting pheromone redundancy. A live queen is probably sending a message through multiple pheromones produced by multiple different glands. Essentially, we've shown that in this experiment, the pesticide exposure during queen development did impact the queen physiology by changing the mandibular gland components. And this in turn impacted worker behavior. So now let's move to the sexy portion of the dissertation research that I'll share today. So the overwhelming research question in this, I'm sorry, my neighbors are doing renovations. The overwhelming question of this portion of the research, which was, does pesticide exposure affect queen sperm viability and or mating frequency? So mating frequency is just the number of drones that the queens mate with. And we suspected that pesticide exposure would decrease sperm viability and mating frequency in the queens. So just to give you a little bit of background about these topics, just like you got a little bit of background about pheromones, virgin queens leave their nests to take nuptial flights to drone congregation areas, and there they'll typically mate with about 12 to 16 drones, although this can vary. And the resulting sperm that is kept will be stored inside the queen sperm storage organ, which is called the spermatheca, and it'll be there for the rest of the queen's life. This is one of my favorite beekeeping pictures. You may not be able to tell, but this is my finger in the picture, and a Texan beekeeper named Cameron Crane took this picture. Here you can see three queen spermathecae. The one on the right is from a virgin queen. The tracheal net, which provides oxygen and keeps the sperm healthy, is here. But you see that the spermatheca itself is totally clear. That's because there is no sperm inside of it. These two spermathecae, and you can see the tracheal net of this one here, that these two have this sort of cafe au lait color. And that's because the marbling that you might be able to see in these spermathecae, that is caused by sperm presence. So these are the spermathecae of mated queens. So first I wanted to test sperm viability. During mating, not all sperm is kept, but the sperm that is able to migrate to the queen spermathecae is pretty much always viable initially. Otherwise, it wouldn't have been able to make that trip to the spermatheca. Now, when you dissect a spermatheca and put it into a tube and then rupture it, the spermatozoa, which are these squiggly things, unfurl and they swim all over throughout the mixture. So if you take this mixture and you introduce fluorescent dyes. You can dye viable sperm green with Cyber Green 14, and you can dye non-viable sperm red with propydium iodide. And if you count these up, you can come up with a viability percentage. Here are those results. And you may not be able to tell, but there are no letters or asterisks on this graph. That is because there was no statistically significant difference in the sperm viability of these queens in my study. Now, and we can touch back on that at the end if you're interested. But now let's talk about mating frequency and queens for a second. So in a colony, we have what's known as intra-colony genetic diversity, and you can actually see it in this picture here. You can see where these are all workers from the same mom in the same colony, and you can see that they have different colors. Look at this. This is a very orangey bee. This is a goldy bee. These are darker, blacker bees. These bees had different dads. So they've got what's known as high intra-colony genetic diversity. And this is important to a honeybee colony because the more diverse a colony is, the increase in overwintering success and survival. And there is also a decrease in pest and disease presence. And that was most strikingly shown in work out of Tom Seeley's lab. Now, how do we measure something like intra-colony genetic diversity or mating frequency or the number of drones a queen mates with? For our intents and purposes, these are all interchangeable phrases. Here in the bottom photo, we see purple-eyed pupa. Those are the ones that I took. So after each queen had laid many eggs and the eggs were allowed to get to this purple-eyed stage, I took them and I killed them. And I sound like a serial killer laughing about this, but I guess most biologists are ghouls in some ways. And you kind of have to, in this case, kill a bee if you're going to extract its DNA. And so I extracted the DNA of these queen's offspring, and I examined very, very short repeating sequences in the genome that are known as microsatellites. By doing this, I was able to measure both observed and effective mating frequencies. And so just bear with me one sec while I explain what that is. So the observed mating frequency is just the number of drone fathers that you have evidence for in the sampled offspring. The effective mating frequency is just a calculation that you use that tries to correct for skews and biases. So for instance, here, if I looked at 100 offspring, I might see, well, 33 offspring had one drone as a dad, 33 had drone 2 as a dad, 34 or 30, whatever, had a third drone as a dad. So the observed mating frequency is 3 because there's evidence of 3 dads. And the effective mating frequency is very, very close to 3 because the effective mating frequency, again, is trying to correct for any paternal skews or unequal contributions. In this case here, we've got a scenario where one drone fathered maybe 75 offspring in a sample of 100. And another drone maybe contributed another 20 offspring. And then you have a third drone which contributed maybe 5 offspring. So you have these unequal contributions by the fathers. In this case, the observed mating frequency is 3, but the effective mating frequency tries to give you a correction for this unequal contribution here. And so the effective mating frequency is only 1.58. And I hope that that makes sense. Now, after I took the queen's offspring and extracted DNA, I did PCR, where I copied the relevant bits of the genome that had the short repeating sequences. And I did this with a veritable army of undergraduate students shown here. So Omar Khan, Kyle Zhu, and Charlie Webb really helped with this portion of my project. I analyzed the resulting DNA fragments using a sequencing machine. And this is the sort of output you get from that, where the spikes correspond with the numbers of repeating segments. You put this data into a program called Genius and use another program called Colony, and then all of a sudden, you're able to calculate observed and effective mating frequency. Here, the letters denote statistical significance. And here we do see that there was a difference between the number of drones that control queens and fluvalinate and cumulphos queens and amitraz queens mated with. However, you may notice this is not the difference we expected to see. More drones mated with fluvalinate and cumulphos queens than mated with the control queens, which is very interesting. And these results are echoed in the effective mating frequency, which you see here. So why on earth would this be the case? So mating frequency, just like retinue size, has kind of historically been used as a proxy measurement for queen health in general. Why are we getting two different answers with these different proxy measurements? And why on earth would queens that were reared in pesticides be more attractive to drones than queens that are reared in pesticide-free environments? Well, those are really neat questions. So just to give a short recap, we do know that pesticide exposure impacts the mandibular glands of queens and that this difference is large enough that workers can detect it. We saw that pesticide exposure didn't impact the sperm viability, but we do see that pesticide exposure has impacted the mating frequency. I think that this is the coolest, most weirdest finding in my dissertation. And part of that's because I can't give you a cut and dried answer of why we found what we found. A smaller scale study done by my advisor and her postdoc advisor in 2016 found that control queens did mate more with fewer drones than queens that were reared in one pesticide group. But people kind of thought that this might be a weird data artifact or that this might be a weird geographic quirk or something odd. But this weird finding was reproduced in my dissertation to an even larger extent and with queens that were reared in multiple pesticide groups. This was done in a different geographic area and years later. So this actually probably is a biological phenomena that could be consistent. I have a lot of ideas about why this might be, but this is the portion of my dissertation that would most easily lead to further follow-up studies. And ideas that may explain this would be that perhaps the pesticide exposure damaged the queens. So queens are the ones that choose to end mating flights. maybe the signals in the stretch receptors that say, okay, we've got enough sperm, we can stop. Maybe those stretch receptors aren't working so well, or maybe the signals from those stretch receptors aren't making it to the queen brain. Maybe they just keep flying longer than control queen counterparts. There are other hypotheses or ideas too, but that's the one that I'm kind of interested in. Research that I did, but that we won't talk about tonight, because I do want to talk about a few other things tonight rather than just the PhD research. These findings were not due to differential feeding rates of queen larvae, which was something I pursued in a different chapter. And pesticide exposure did not impact queen morphology. So the size, the general weights of the queens, things like that, that have also traditionally been used to measure queen quality. But again, and I won't get on my soapbox too much, but many of the things that scientists use to measure queen quality are proxy measurements. So things like body weight and morphology, those don't really, those aren't the same as something like egg-laying rates or a tangible impact of the queen's health. We tend to use proxy measurements for a lot of reasons, but this dissertation work certainly brings up some interesting questions about doing that. This work was possible because of a lot of support, both from my advisor, Juliana Rangel, and my committee members, in addition to my lab mates, various mentors, and support from a lot of different organizations. So, how does this apply to us as beekeepers? You may not have been able to tell from the talk so far, but I really love giving context and background. So we're going to do that for a second. So I'm from the US, which Renata said in her introduction, and I grew up in Wisconsin. So you may not know, but Varroa mites were first detected in Wisconsin in September of 1987. My good friend Gary Oreskovic, who's a retired beekeeper now, was actually the one to find them. And by good friend, I mean he yelled at me at least twice when I worked at the beekeeping supply store, but I hold a lot of fondness for him. And I think he does for me too. Gary was absolutely disturbed and freaked out and concerned and all of the things when he found Varroa in his colonies. And part of that was because at the time, Gary migrated from Florida to Wisconsin and back again. And so Gary was very concerned that he had trucked bees with this new crazy parasite halfway across the country. And so he sent a sample of bees that have Varroa mites on them to the USDA, and he went through his entire operation, which I think was about 3,000 colonies at the time. We didn't have the sampling techniques then that we do now. So he was visually inspecting bees in all of these colonies. And he found Varroa mites visually on bees in about 19 of his colonies. And he was so concerned that he burned every single one of those colonies. The USDA confirmed his find the following month in October. And 3 days later, the EPA licensed the use of fluvalinate, a miticide, in the US. Fluvalinate, although I neglected to say it initially, was the active ingredient in apistan. Later that same month, Varroa mites were found in Florida, which indicated that Gary had taken them from Florida to Wisconsin earlier that summer. By January, Varroa had been found in 10 different states. Shortly thereafter, Amitraz was first licensed for use, although 2 years after it was licensed for use in 1991, Amitraz lapsed, the license lapsed because beekeepers were complaining that it caused their queens to stop laying. And so the company that was using it did not pursue a renewal. So we really only had, as a beekeeping community, fluvalinate for quite some time. And so when Varroa mites showed resistance to fluvalinate, which was documented in 1994, it really caused a lot of problems. Beekeepers began to double the dosage rate of fluvalinate and to use things that were not legal to use in their colonies, including using Amitraz again. By 1999, a new miticide, Coumaphos, entered the market. Coumaphos was the active ingredient in Checkmite Plus, if that rings any older beekeepers' ears. By 2001, there were already documented Varroa mite groups that were resistant to Coumaphos. And by 2005, there were Varroa mites that were resistant to Amitraz. I would like to especially point out here that Amitraz was not legal for use from 1991 to 2005, but because it was still being used illegally, Varroa mites were showing some resistance. Despite this, they were showing more susceptibility than they were to fluvalinate and Coumaphos. And so in 2013, Amitraz was re-registered for use in the US, and it's still used today in both the US and Canada. Now I want to introduce you to the idea of the chemical treadmill. If you haven't heard of this before, it's the idea that in a crop system, be it crop or livestock system, pesticides are pretty much solely relied on to fight a pest or a pathogen. And this is a problem because a hallmark of the chemical treadmill is that resistance in the target pest is common. And this leads to the necessary development of new pesticides to replace newly ineffective ones. Do you think that we as beekeepers are on the chemical treadmill? Because I do. And I hope that the timeline I laid out a little bit earlier gives you some evidence that backs my case. So what do we do in result of this? Well, we utilize integrated pest management studies as strategies where we can. So what is IPM, or integrated pest management? Integrated pest management is an idea that entomologists and ecologists have actively campaigned for since around the 1970s, when we realized how prevalent and prevalent the pesticides we use are in the environment. So the integrated pest management pyramid is shown below, and the idea is that you use a variety of practices to suppress a pest population below an economic injury level. Something to emphasize here is that we're looking at pest suppression, not eradication. IPM centers around this idea that some variation in your pest population is going to happen, and that eradication probably isn't feasible in many cases. If you use IPM, you're trying to maximize the benefits of various pest management strategies while minimizing the costs. And these costs can be temporal or economical or any sorts of things. So in beekeeping, well, in all IPM strategies, you work from the base of the pyramid to the top. And so in beekeeping, a cultural control that you would start at would be utilizing some sort of resistant bee stock. So bees that exhibit high hygienic behavior assay scores and things like that that might combat Varroa or whatever pest you're looking at managing on their own without human intervention. That would be an example of a cultural control in the bee yard. If that's not cutting it, then you move up a step on the pyramid to physical mechanical controls. And an example of that would be things like traps. So things like drone comb trapping for mites, that would be an example of a physical mechanical control. If that's not working, along with cultural controls to control your pest population, then again, you move a step up the pyramid to any biological controls. So some people espouse the idea of a predatory mite that will eat Varroa mites. That would be an example of a biological control. If that's not cutting it with physical mechanical methods or cultural methods, or if that biological control method doesn't exist, then you move up a step on the pyramid again to what we commonly refer to as soft chemicals. So things like oxalic and formic acid or thymol-based products like Apiguard or other essential oils. If all of those still aren't working, then you take that last step to the tip of the pyramid to synthetic or as the table here calls it, conventional pesticides. So things like amitraz would fall into this category. Now, I'm not sure if you remember that blue timeline, but fluvalinate, the active ingredient in apistan, was registered for use 3 days after the USDA confirmed the find. And this is astounding to me. But essentially, as beekeepers, we've gone from the top of the pyramid to the bottom. We haven't gone from the bottom to the top. And this is true in Canada too. Now, how do you know when you have to take a step up the pyramid? Well, you sample. You sample for your Varroa mites. You find out if you need to move up a step on the pyramid. And you sample more and you sample more and more and more and more. There's never enough sampling in an operation that really utilizes IPM strategies. All right, and I have one little bit of soapboxing before I'm gonna quick tell you what I'm up to now. So as beekeepers, it's kind of important to be neighborly. So you can control your Varroa mite levels, and that's great. But if your neighbors don't, then you've got Varroa bombs in the area, and they're going to affect you and your operation. Likewise, if you're not controlling your Varroa mite levels, you're going to impact the apiaries around you. And I am very excited someday to meet you all in person. But for the intents and purposes, I'm assuming some of my audience are hobbyists and some of you are commercial beekeepers, and I hope that that's true. I'm also assuming that some of you have been beekeeping for a very long time, and some of you haven't been. So I also hope that's true. But the idea here is just to keep an eye on your Varroa mite levels so that you can be neighborly and keep your Varroa mites under control. And part of the reason I say this is because a lot of hobbyists seem to get enamored with the idea of going chemical free, which I think is a really great idea that is incredibly difficult to put into practice in beekeeping. And if you're practicing this sort of Darwinian beekeeping where you don't put any pesticides in your colonies, regardless of your pest levels, then you could be setting your neighbors up for quite a bit of disaster. Part of this is also in your definitions. So keep in mind the difference between a soft chemical and a synthetic chemical. Similarly, you have beekeepers that take varroa mites very, very seriously and that don't sample their varroa mite levels before they treat. I would label this as prophylactic treaters. And I hope that some of my dissertation research shows why this is not a good thing either. So the residues that Mullen et al. Survey found were in part shocking because fluvalinate and coumaphos were there in 98% of the samples in 2010. Think about that timeline. Neither Apistan nor Checkmate Plus had been used by beekeepers for years at that point. But the reason that they're there in the wax is because they have a long half-life in beeswax. And so you get miticide residues in the beeswax, even if you've never used those products before, because as beekeepers, we sell our wax. We sell it to beekeeping companies who make foundation out of it. And then new beekeepers that have never heard of these products get foundation that has residues of fluvalinate and coumaphos, among other things. Now, why does all of this matter to you? Well, if we're on the chemical treadmill, that's something we need to be aware of so we can take steps to address it. We've seen in my dissertation research that there are sublethal effects of these miticides that we use. They're definitely affecting our queen health in at least some ways, especially if you look at the egg laying rates. That's something that's worth paying attention to. So I would say that honestly, as an industry, our best method of controlling pests like the varroa mite is to utilize integrated pest management strategies. At the time I was conceptualizing my dissertation research, there was a relatively low number of honeybee pesticide studies that were using field relevant rates of exposure. And so it's interesting to me that the field relevant rates of exposure showed that the miticides had a huge impact on queen health, but the agriculturally applied pesticides didn't really have that big of an impact. My research really indicates that the developmental pesticide exposure affected queen physiology and worker behavior. And I do want to end this portion of the talk with just a little FYI. Research is almost always a game of catch up to the real world, catch up with this new and emerging pest or disease or problem. And in this case, with pesticide research in honeybees, the game is kind of stacked in the favor of no one when unregistered pesticides are being used in apiaries, like amatraz was after it lapsed registration and things like that. So what about your operation? Well, size matters in the context of apiary operations. We see in national surveys in the US and in Canada that small beekeeping operations tend to have very high loss associated with not using miticides and not adequately controlling the varroa mite levels. But we also see that large beekeeping operations tend to overuse miticides. So in honor of Halloween last month, It would be a good thing to find a happy medium here. And I think that that would best be done by incorporating a lot of Varroa sampling into your operation and making those educated choices about how to deal with the Varroa mites based on that. We also kind of have to play the concentration game because miticide residues are found so ubiquitously. It's going to be incredibly difficult to find beeswax that's pure anywhere on the continent. For the beeswax that I used for the control queens, we actually sourced beeswax from Africa because it was the only beeswax that was pesticide free. Now, I'm going to end our time together just chatting a little bit about what I'm doing now. So right now, I'm a postdoctoral research fellow in Dr. Steve Pernall's lab at the Beaverlodge Research Farm. And here's a picture of the Beaverlodge beaver because I just get a huge kick out of it. I'm primarily working on a national project called BCSI, where we hope to develop a next generation diagnostic tool for you all to use here in Canada. So in order to develop this tool, we're looking at things that are stressing and killing the bees and seeing how these things impact the microbiota, gene expression, and protein profiles of nurse bees. This project is done in collaboration with over, I think 11 exactly, but I should double check that, but with many other honeybee research labs throughout Canada. Oh, and here's a picture of me with our sign. Okay, so here's a picture of me and Michael Pearson and Abdullah Ibrahim, which several of you know. Our lab is working on a lot of honeybee disease ecology portions of the BCSI project. So working on AFB and chalk brood and nosema stuff, among others. And here's a picture of the AFB. We're also doing some pollination stressor work. So here we have a picture of our colonies by a canola field. Now, what can you do to help specifically, if I may be so bold? We have 2 BCSI surveys out right now that you can take, and that I'll drop into the chat, or drop the links to into the chat. One is a COVID impact on beekeeping survey, and that one's going to close quite soon, actually. I think we're planning on closing that on December 1st. The other survey is going to be open quite a bit longer, and it's on the diagnostic tool front to better suit Canadian beekeepers' needs. Alberta beekeepers haven't participated a lot in the survey yet, so please, please do. It would be great for our province to be represented so that it's not just beekeepers from Quebec or Ontario or 1 or 2 other provinces. So I think that's all. I've listed my email below. You're welcome to contact me with questions or comments if you don't want to say them now. But I would love to have a Q&A session if we've got time. I notice I'm slightly over time, so thanks so much for your patience and interest. I really appreciate it.
Thank you very much, Liz. So much good information. It was very, very interesting. I have a few questions, but I would like to maybe explain a little bit how the Q&A works in a webinar because everybody's muted. If you'd like to ask your question verbally, you can click on the little icon that says raise your hand, and you can raise your hand and I can unmute you. Otherwise, you can use the chat box to ask your question, and Liz can see it as well. So while we wait to see if someone has question, I'll ask mine. So this is very interesting about the retinue size. I started doing some queen work this year, and I took a few queens from my queen bank and I sent to the National Bee Diagnostic Center to get the sperm viability and ovary size and all of that measured. So I have an idea on how they looked before I start the project. And very interesting, Patricia at the National Bee Diagnostic Center, she said when she received the queens, she noticed that one of the queens in the cage was very um, excluded she was being bullied. None of the workers that I had added to the box, I had quite a few, was with that queen. And so she made a little mark. And when she was assessing that queen, she noticed that that queen had the poorest assessments for everything. So one question that I have with that is that, so you notice that yours, for example, had retinue, the retinue side was smaller. Do you know, or did you measure if those queens also have higher supersedure rate or something like that? Do they get rejected as well in a colony?
That would have been, I did not, but that would have been such a cool study to do. And I do think that I'm super interested to hear that the things that Patricia used to assess, like the sperm viability and the ovary size, that those correlated, because that's good to hear. I'm always interested in what things correlate and what things don't, because it seems like there's a lot of inconsistency sometimes. And I don't know why, but it would be really neat to find out.
Yeah. Okay. Well, be interesting if someone looks at supersedure rate and if they get accepted or not, because we do. There's quite a few queens when you order and you introduce to your hive, and the hive doesn't take it. And a lot of times, many beekeepers will say if the hive didn't take it, it's for a reason, right? And we never know why. So that it would be interesting to if things correlate with queen mandibular pheromone, so maybe do those queens have lower pheromone or something like that, and that's why the hive rejected or something like that, right? So be interesting. I know you have a question here, so I will save my second one. Someone asked you, why did you use beeswax with JZBZ cups?
Yeah. So I first wanted to double check that there weren't, okay, let me start over. I coded the cups because that's what some of the beekeepers were trying to do in Texas, including the ones around me. They were coating JZBZ cups in beeswax or putting beeswax along the edges of the cups because they felt it really increased their takes initially and their takes to emergence. And so that's why I initially did that. And then by the time I thought about not doing that, I was already partway through the experiment. So you can't change a variable like that halfway through.
But you used pesticide-free wax, you said, right? Okay, yeah.
So for the control queens, it was pesticide-free beeswax, and for the experimental queens, it was.
They were treated.
Mm-hmm.
Yeah.
And I didn't talk too much about why I paired the pesticides that I did and why I left amitraz separate. But the reason for that is because fluvalinate and coumaphos were found together in the vast majority of wax sampled, whereas amitraz was found more infrequently compared to fluvalinate and coumaphos. And then chlorothalonil and chlorpyrifos were also found together fairly frequently.
Okay. Yeah, so my next question is kind of on that line. So nice that you brought it up. But so because we do find, well, fluvalinate coumaphos is found, it's very prevalent. We find it in pretty much every, not every, but it's very common. And now with our use of Amitraz, we also find very common all of these pesticides that you use in the same colony, the same wax. Did you think about maybe doing a treatment, all the combined to a big soup? And what do you think would that maybe show? Or
It would
anyone
be,
has done?
yeah, it would be really neat to do that. And so after I started this work, Joe Malone, who just graduated out of David Tarpey's lab very recently, he was looking at something kind of along those lines, where instead of pairing pesticides based on frequency in the wax like I did, he was looking at hazard quotients and combining pesticides until they reached a certain hazard quotient and trying to rear queens in that. Those papers aren't out yet, but what I know from seeing his talks are that the bees didn't like to rear queens in the most pesticide-laden beeswax. So
Yeah, no, that's very cool. So
well,
I guess go ahead.
no, I sort of have one more, like a follow-up comment, and that would be that it's kind of like what you said with bees that may not accept a queen, where it's like they know what they want and what they don't. And sometimes we know what criteria they're choosing, or we think we know, and other times we don't. And so the bees are choosing when they have a choice, at least in Joe's study and mine too, to some extent, that the workers don't like rearing queens in pesticide contaminated beeswax. They'll choose not to if they're given choices. So that's really neat.
Yeah, and I mean, especially with your retinue size data, that shows really well that it changes the bee's behavior, and that may change the queen behavior as well, because you're not getting the attendance that she needs, right?
Yeah.
No, that's very, very interesting. Tracy's asking a question. She said, I read recently from Steve Pernau and Johan van den Hever that DCH from Fumidel B accumulates in wax. Any thoughts on whether that may affect queens? Great question.
Ooh. Yeah, that's the most accurate answer is that I don't know. It would be neat to find out. A lot of residues in beeswax have the potential to impact queens if the queens are being reared in that wax, or workers or drones if they're being reared in that wax. But I don't specifically know if DCH would.
Yeah. We know formic acid. We have studies that have shown people say that they don't like use formic acid with in colonies because that affects their queens. Did you look at anything like that as well? I'm just kind of following up on what Tracy asked, and I'm curious if you have any thoughts on formic acid.
Yeah, I haven't specifically done research with formic acid, but in my seeing it used, I can tell you that at least when I've seen it used, it can impact queen behavior. So for instance, in Wisconsin, a common complaint is that when you initially treat a colony with oxalic or formic, if the queen was laying a little bit, she stops immediately. And so you got to time your applications, because if she stops laying at the end of the season, okay, maybe that's a good thing. But if you've timed that application not perfectly in a time where you would want the queen to be laying eggs, then that might cause problems.
Yeah,
I don't know if that answered your question or not, Renata.
No, it did. It did. Some people, yeah, there is a temperature requirement, but yeah, some people might use it a little earlier or use it in the spring, right as the spring treatment. Um, no, that's great. Because I don't see anyone asking question, I'm going to ask another question. So you said that you measured queen morphology. Did you also measure ovary size and ovary volume? You did, okay. And that
Yeah,
was
so
not different.
yeah, so non-significant ovarial counts. So, and that paper is not out yet, but it will be someday.
Okay.
Yeah, the other two chapters have papers out. And if people want to read them, please just shoot me an email. I'll put my email in the chat too, and I can send the papers to you. Although I do have to say that if you ever thought that the talk itself may have been dry, you'll find the paper particularly dry, but that doesn't mean that you shouldn't have it if you want.
So my last question, because a lot of beekeepers will have their equipment for so long in a colony, and that means that more pesticides could be accumulated in that wax, right? What is your recommendation then would be for commercial beekeepers and hobby beekeepers both? What do you think would be the best? I guess maybe for commercial, because they have so many, the economical, it might be more expensive. Maybe you would say you guys can leave for a little bit longer, but what would be your recommendation for changing your equipment? How much percentage, like how many years you would say, kind of thing?
Yeah, so to play that concentration game, yeah, of concentrations of pesticides in your wax and keeping those concentrations as low as possible. For commercial beekeepers, I have heard varying recommendations of how many frames to cull out per year, but for the biggest beekeepers, 10% of frames per year. So like the 10% of your oldest frames is kind of what people have recommended. If you have the economic means, I've seen people, at least on like 5-year bases, cull out up to 30% per year of their oldest frames. And so a lot of this is just what you can afford, how old your frames are to start with. So if your frames are super, super old and you've never culled out frames, then it might be a good idea to cull out a high percentage as soon as you can, and then you can step back. Yeah.
Yeah, and I guess it would depend on your IPM program to how heavy do you rely on those miticides, and maybe where your hives are located, they near maybe fields that might be a lot of pesticides.
Yeah, well, and we haven't talked about it tonight, but the presence of the miticide residues in the beeswax really, really do contribute to Varroa forming resistance to those products. So if you're somebody that really leans on miticides, then you want to be incredibly aggressive about culling out the frames for a lot of different reasons. for your bee health reasons, but also because it would be devastating if your Varroa mites were to become resistant.
Yeah, yeah. Yeah, someone raised a hand, I think was T.R. Barnes, but the hand is not up anymore. Maybe it was a mistake. But if you was not, raise your hand again and I can allow you to talk.
Oh, Craig T says they call 20 to 30%. Good for you.
Oh, wow, nice.
Yeah.
Yeah, that's a good goal.
Yeah. Yeah. I think so then, Craig, if you don't mind me asking, I'm assuming you keep track by writing on top of the bars what year they entered your operation. Is that correct or no? And no worries if you have stage fright.
Yeah, that's a good number, 20 to 30%. I know some beekeepers that do around 10 and some that do on the commercial side that do around 15 to 20.
Yeah.
But
Plus,
we do
yeah.
write on top.
Well, and especially when you have those rough years, some years it's hard to cull any much, like you do what you can afford.
Yeah, exactly. Especially you're like we have been having this year and last year, but yeah. Well,
So Craig did reply, they do write on top, and their hope is that every 5 years there's a full changeover.
yeah,
that's a good system.
Nice. Well, I don't want to keep you too long. I really liked to hear more about all your PhD research and your findings and things like this. I think we all need to get better at managing our own pesticides, the pesticides that we apply to the hive, because that's a good chunk of pesticides that we introduce every single year to the colonies, and we need to be more mindful that those who stay in a colony, they stay in the wax, and the more we use that same wax, the more pesticides are going to be in there. And it's nice to see the effects and the detrimental effects of having waxes that is full of pesticides, and that we I know it's expensive, but we need to do something about it, right? So that's very good. Thank you very much for your time, and thank you for all the good research you did and for presenting it to us. Thank you.
Thank you so much for the opportunity. I really appreciate it.
And welcome to Canada.
Thank you. I really can't wait to meet you guys in person.
Yes. You arrived in an interesting year, but hopefully very soon we'll be able to meet in person again, and it would be nice to have you in person, giving a talk and meeting all the Alberta beekeepers. Thank you, Liz.
Very much, guys, for your time and attention.
Have a good night.
Yeah,
Bye.
you too. Bye.